Does that work for you folks?
Sounds good.
All righty. All right, so chapter 13, we're looking at memory. And truthfully, one of the, well, I sort of have a little soapbox about this, because we hear a lot about learning disabilities and, processing disorders and stuff like that. And I always sort of fall back on the idea that a lot of that is really guesswork. And it's not really that scientific, in terms of understanding how learning disabilities work. It's primarily learning disabilities are interferences with memory, and somewhere in the process of memory, either in the perception of stimuli, the entry into short-term memory, the processing or the organization skills that somebody has, or distractions that come along that interfere with attention, all of these processes that interfere in the memory cycle. So we can pinpoint sometimes when those things, based on what we observe in a person's behavior, we can sometimes pinpoint where they might be having some trouble. But at a most fundamental level, in terms of the brain, we don't even know how a memory is made. I mean, I think this is probably one of the best examples of why it's so cool to study psychology, is that there's so much we don't know. There's a lot we know, but there's so much about the brain that we don't know, and it's infinitely complex. And, and I watch, science shows, and they talk about building computers that will rival the human brain. And it's like, we're a long way from that. You might be able to rival, you could do math faster, you could do a lot of things faster, and we like that. But I was talking to the daughter of the keyboard player in my band is a robotics engineer. And she said that, I was saying, what would it take to create a robot that'd be able to, because I had just done it that day, to look at the dishwasher, take my hand like this, reach in and grab 3 plates at the same time and pick them up and put them in, take them out of the dishwasher. And she said, we don't even have that yet. Unless we had the dishes perfectly straight, and, you could, of course, create a machine to do that, but to actually kind of visualize moving my fingers around to get a grip, to sense, that I've got them all in the right place past the knuckle so I can get a grip on them and pull them. So there's quite a gap between that and the human brain. And here's this chapter talking about memory and what we do know about memory and what we think might be going on when it comes to memory. So the chapter begins with, let me get into the course book here. The chapter begins with just looking at the 2 major theories of memory, which I'm sure you were exposed to somewhere along the line, probably in Intro to Psych. You have classical conditioning, which is learning by association. So as in the experiments that Pavlov did with his dogs, speaking of dogs, we just had a conversation of dogs there. So here's the dog that has learned that if he licks Jaden's face long enough, he'll get patted, and that's becomes associated. And I'm sure, Jaden, and whenever you go anywhere near the food, the dog's right there thinking it's, food time. And they don't need to have processed that information very much. That's just association. You're close to that, I must be getting fed. And then there's Skinner's model of operant conditioning, which is reward-based. We're more likely to do things when we're rewarded for them. Now, in biopsych, just like I said at the beginning of the class, the emphasis is, can we explain all of human behavior biologically? Can we look at the brain and see what's going on in there? And we're really, it's interesting because we'll talk about this a little bit on Wednesday. It's all about, it's really just a correlation between what we observe in the brain, things that we see in the brain that seem to be associated with things that we see in terms of, memory production, but we don't know the cause effect. And so almost everything we know about the brain and memory is a correlation. We see changes in the brain and they seem to happen at the same time as memory processes, and we figure there's something going on there. Now the textbook and the course book go into this thing called an engram, and I wanted to give you a notion of what that means, and that is it's a theoretical a structure or group of structures within the brain that are responsible for a particular activity. And it's not necessarily like, oh, there's the memory part of the brain. We have the lobes of the brain and we have all the different processing areas of the brain and stuff. But as we've been discovering in this class, you might have started this class thinking, well, visual information is processed in the occipital lobe. Now that is not all that the whole brain gets in on it. And its information is processed, when we looked at the different pathways for all of our information, that the entire brain is involved in seeing things and the entire brain is involved in remembering things. But we kind of look for that set of structures that we think are pretty important, or a set of chemical processes that we think are pretty important that are related to memory. And the one I'm most familiar with, the other engram that I'm most familiar with, and you might have heard about it if you took developmental psych, and that is in the process of language development. And language development, there's a guy named Noam Chomsky, who's a linguist, and he developed a model called the language acquisition device, the LAD. Now that LAD is an engram, meaning it's just that one is the theoretical parts of the brain that are involved in in language development. So he recognized that there were some aspects of language development that were universal, that kids learn syntax. That's the ordering of words, and in particular to the language, they learn that, and they learn parts of grammar, like when you add ed at the end of something, it refers to the past tense. Like I talk now and I talked yesterday. And they learn those little clues for manipulating language to produce a meaning. And they learn the rules and apply the rules in ways you can tell you can demark periods of time when in the language development of a child, when they've learned certain rules, because they actually start making mistakes and they produce language that they would have never heard out loud because they're applying the rule. And the example that's often used is a young child, maybe 3 or 4 years old, will say, I falled, F-A-L-L-E-D, I falled down yesterday. And what they did in the English language, there's another word for the past tense of fall, which is fell, but talked, walked, all of those words had those EDs at the end. So they learned the rule to put an ED on the end, but then they misapply it. And then we're like, there we go. They learned the rule and they actually dynamically produced this word that actually is not a real word. So that language acquisition device that has identified parts of the brain, mostly in the temporal lobes, associated with language production, language understanding, be able to name objects, you know, the Wernicke's area, Broca's area, those things, that we have that's sort of an engram. We are even further away from an engram when it comes to memory, because certainly memory is almost like a cognitive universal. I just made that up. That's a kind of neat word, cognitive, nice phrase. Cognitive universal. Memory is involved in all of our cognitive processes, including language. So in order for me to identify this as a spoon, I have to be able to remember and recall and store that information, see a spoon. Also, interestingly enough, that, you know, when we talk about memory, this is how much of an object do I need to see before I can successfully identify it as a spoon? How much of the shape needs to be there before I start applying? So the memory is so good and it's so cognitively based that we can take clues. And there's actually some experiments they do. That'd be actually kind of a cool experiment to do. It'd be there's drawings that you can get that successively approximate the full drawing of an object, and how much data do we need before we recognize it? And because we fill in the spaces, you know, and kind of like, oh, it's starting to look like this. I almost like to connect the dots. Thing. You as adults, for example, can probably open up a connect the dot book, and you don't have to connect the dots. You can see what's there. You can see what the object is. You can actually trace the lines inside it in your mind. But that has to do with drawing from memory, being able to, you know, place that upon the page that's right in front of you. So in the history of psychology, there's been this search, you know, what are the parts of the brain that are associated? What's the biology of memory? And we see some specific parts of the brain here that are identified in our text as being important to memory function. The lateral interpositus nucleus, not a mouthful, not my favorite part. It's important, and it's a part of the cerebellum that's associated with memory. Then we have the hippocampus, which is probably the part of the brain that's most associated with memory. It's the as I got into the book, it's sort of shaped like a seahorse. That's how it got its name. And this is we mostly know about the hippocampus because of people who have experienced strokes in that area. And we don't know until after they've died sometimes. I mean, certainly with the history of it, sometimes we can identify the area of the stroke through an MRI or something like that. But when people have had damage to their hippocampus, there's usually some pretty significant memory problems that they have. And I included a movie in there about case studies that relate to that. And it's amazing. I mean, there's I'm not sure if it's in that particular movie, but I remember watching one where there was a guy who would wake up every day, and it's like Groundhog Day, you know, it's the same day every day that he's waking up, and he has no memory of anything. He does have a memory of his own identity and his age, and waking up, he would wake up, and after a while, he became so distressed because every time he saw himself in the mirror, he would see himself as older than his internal representation. So in the group home that probably not even a group home, in the apartment that he's in, the staff removed all of the mirrors because he was just freaking out every time. Our identity is based in our memory. It's like what I was doing yesterday that made me do something today, and what I'm expecting to do tomorrow. Our entire sense of self is based in memory. So very important parts of the brain here. So it goes a little bit into hippocampal damage, basal ganglia, prefrontal cortex, the amygdala, each one of these sort of, and the testing I'm giving you on this is pretty straightforward. It's just matching parts of the brain that we think are associated with memory. The last part is looking at this at a cellular level to see, is there evidence in the structure of the brain at the cellular level that learning is happening? And kind of the only thing we have going on right now is this sense of, and if we look at the graphic here that I included, that there's a process called long-term potentiation. And that means, for lack of better ways of thinking about this, you can think about it as a trail in the woods. And the more you go through the trail in the woods, the more worn that trail becomes. So when we are learning something and we're passing the same signal through the same set of neurons, the path gets worn. So it becomes a familiar path. And it becomes easier to walk. And just like when we learn something new, it's kind of difficult and the branches are in the way. But then if we keep doing it over and over, eventually it becomes easier to do, and the path is clear. It also can be represented by the fact that if we don't go down the path, sometimes the path gets overgrown again, and it's hard to do it when we haven't done it for a while. So the analogy kind of carries like that. But what does it mean to wear down a path? I mean, it's a nice analogy, but what does it actually mean in terms of the physical brain? And what it means is these chemicals here, that when a bunch of neurons are being worked over and over, as in learning, as in the rehearsal, let's say, process of learning. Let's say you're learning a song on guitar and you play it over and over. In the process, all the neurology that's being involved in the brain and in the body of learning how to do those things, that there's a particular kind of receptor that gets produced, and that receptor creates a cell that has become sensitive to stimulation of the particular kind. And so you can, maybe you can think about it like if we think about muscle memory. Has either one of you ever driven a standard? You drive a standard? Okay, Jaden, do you drive a standard?
No, I've tried.
Yeah, it's not easy. You're thinking, when you're driving it, I'm actually sitting here doing my pedals as I do this. You have the clutch, and then you're changing gear, and then it's like, how much gas you put in, and your foot's on the brake, and it's all this kind of coordination. And it takes a while before that becomes automatic. But you think about, you're going down that trail, and what they're thinking is, in the brain, what they do actually observe is this series of neurons that is involved in those actions become sensitive to working together. So over time, it doesn't take a whole lot of stimulus for that whole process to go through, and it automates, and people become really used to it, and they can do it very quickly, and they don't have to think about it. Unlike the first or second, third, maybe 15th time you stood it, you were in a standard vehicle and you still couldn't get it. You're still working like, which one do I go with here? But after a while, it could become automatic. Like, it becomes so automatic that we can, take that person, like I am myself, my car is a standard, my wife's truck is a automatic. And when I sit I actually purposefully, if I'm using her truck to do something, I sit in the car for a little bit, and I remind myself it's not a standard, because that brake is right where the clutch is. And my automatic, if I have to stop in an emergency, my automatic is, of course, my left foot pushes the clutch and my right foot then touches the brake. And if I slam on that brake, usually on the way to the dump. All that stuff will come flying forward. So, so that automatic processes, and it happens, what I described in the course book was physiological changes can happen in the brain that are right here. It's after all of this long-term potentiation, like we're just using them, the neurons are becoming sensitive to firing in sequence. One produces the just like neurons do, they produce the, once one is going, it's very likely the rest are going to go, almost like a domino effect. And what can happen physiologically is the dendrite can build more AMPA receptors, which is that particular kind of receptor, along with the NMDA receptors. They can make more branches. The actual neurons can branch out, and some receptors become more sensitive. So we see that physiologically happening. They can measure all of these things, and they think that maybe that might be long-term memory. Sketchy at best, but you can see the chemistry is there, the physiology there. And even with, even on a kind of simple level, when we look at a brain of a young child, and then we look at a brain of that same child when they're an adolescent, there are way more branches in their neurons interconnecting. Not necessarily more neurons, just more connections. And that goes all the way through adolescence. And then there's a process that we talk about in developmental psych called synaptic pruning, which is truly the use it or lose it mentality of, if you don't do something for a while, let's say you've learned how to use a computer program of some kind. And well, an example of that, years ago, I had this computer, a very, very bizarre computer. I lived the history of home computing because I had all these very weird computers. I had a computer that actually the programs ran off of cassettes, little audio cassettes. You put those in, and there was two of them. One was your program and one was your files, and you put them, just looked like an old tape deck, which I'm not even sure you even have seen a tape deck. Little reel-to-reel cassettes. And anyway, I had a printer associated with that, and I needed to print something out, so the necessity to print was really important to me. And I remember getting this computer, and I couldn't get the printer to work. And I spent like probably 2 days trying to figure it out, and I found some bizarre combination of activities that I could finally get it to print. And so that by doing that, I learned about how the computer works. And I mean, it was really weird. Like, I had to take a tape out, put it back in, press the key F. I had to dance around, go to church, whatever it is I needed to do to make that thing print. And I remember years later that I still had it in storage. I pulled it out and I hooked it all up. It looked ancient. It was. I'm like, I wonder if I could still remember how to get it to print. No way. I know I didn't. When I was using it, it was automatic. But as soon as I didn't have to walk down that path anymore, it quickly overgrew. And probably within my brain, that pathway, because it wasn't being used, the brain is very efficient on energy use, probably the most efficient machine in existence. It's not going to send energy and food to support cells that it's not using, so it kills them off. It slices and gets rid of those. You don't need those anymore. And I guess if I had to learn how to do it, I could probably figure that out again, but luckily I don't. Okay, heading into the discussions and the quiz. The discussion has to do with the ethical question of performance drugs. There was, I'll share that my daughter was diagnosed with attention deficit disorder very early on, and I was kind of skeptical about it. I think she was just acting out. Hard to tell. And of course, this is in a divorce situation. So me and mom, we were getting along and co-parenting just fine, but we weren't always in agreement on this. And so she got her onto Adderall, and I was contesting it. So I went to the doctor, and I, with my ex and Mara, and why is she on this? Goes, well, ADHD is hard to diagnose and stuff like that. And she said, well, just consider it a performance drug. I said, oh, do you give it to all your kids too? I guess it's not a performance drug. But anyway, the notion being that there are medications out there that we give to individuals who may have attention deficit. If we give those same drugs to a person who does not have attention deficit, and it increases their attention, their ability to attend, which is a huge part of the memory process, is that right? Do we do that? Do we take those drugs and do performance enhancement? Oftentimes we create drugs to fix problems, but then we start thinking about, well, what if we use them to enhance performance? That in sports, they have drugs to enhance performance, and they're illegal. So, but what if that came out in the, what if, and the question to discuss is, what if all the rich kids could afford this, and they went to school and they're outperforming everybody in school and getting all the scholarships and getting all the breaks and stuff like that because they could get this drug. Now, these drugs are not that finely tuned, and a drug such as Adderall is a very, what we would call a blunt instrument, meaning drugs are not targeting, they're washing out all over the body, and they have somewhat of a desired effect, and they're not the same for everybody. But as we design drugs more and more efficiently, who knows? So getting into a discussion about that, what do you think about the ethics associated with that? And then the quiz is just, it's a matching of a bunch of terms from, and I like this chapter because as complex as this book has been, and I'm sure I'm going to assume you're reading the book, at least skimming through it. You read that book and it's immensely complex. I don't know if you've encountered the author talks about the complexity and says, you might want to read this chapter twice or something like that. But we still don't know so much. And this chapter sort of really emphasizes, like, here's all the stuff that we have, and yet it's all still correlation. It's not causation yet. We haven't been able to look into the brain in any way and say, hey, there's my memory of the ice rink when I was 5, when I fell and I hurt my head. Can't do it. And then there's stories of people who have had brain damage in areas that presumably have stored memories, and they went a very long time without those memories, and then the memories came back, and we have to ask ourselves, where were they? Where did they go? And I heard one, I have to find this article because I mention it pretty frequently, and it was years ago. There was a theory that was going on that memories may actually be stored, some memories or memory processes might be stored in the electromagnetic field that our brain produces around our head. So in the active electronic field, and the person was actually talking about, well, maybe those people who are psychic can actually sense other people's electromagnetic waves. Who knows? I could see Jaden's thinking, that's what I'm doing. That's why I could read those kids' mind. I know when they're going to throw that ball. And so, okay, so that's our chapter. No labs, no more labs. There are no more labs for this semester. The last one is lab 12. We have, there is very little of the semester left. You're probably recognizing that next week we have no class. It's Thanksgiving. And then we have 2 more weeks after that. And so we're going to be doing just a couple more chapters here. So any questions about biopsych before we look at the lab? Okay. All right. So open up your mics. I want to hear what's going on with the labs. What's happening with, what are you having trouble with?
You go ahead.
Go ahead, Jaden. You were the first one to reach out to me, so.
Yeah, because I like to do the labs like right after, like at least look at them right after we talk about it, so I know if I have questions. But I could get everything set up, like I understood where to put the electrodes and I understood how to record it. But like once I was recording, I had no idea how to like move the red lines to get the certain concepts, like
the different measures.
Yeah, like the different waves and stuff. It said it on the, I know, yes, it had it on the instructions and stuff, but they were like not very clear to me.
Yeah, and you kind of have to like scan the screen. Like I redid the lab for myself this morning to refine all of it, and I had to look around to find the place where something was, and it wasn't as clear. I knew it was there somewhere because I'd done the lab before, but the Jordan, is that what you ran into? Did you get it? You got your heart rate and all that stuff, but you didn't know how to?
Yeah, I already did the experiment. I just don't know how to get like the data.
Okay.
That it's looking for.
All right, so let me bring this up. I have my. Let me go ahead and share my screen. All right. There's my heart rate.
See, mine doesn't look like that.
Mine does not either.
So I'm wondering if we like missed a step. Because like the bottom for the heart rate, mine does not look like that.
Mine either.
Now, this is after, you see this button that has the 2 mountains on it?
Mm-hmm.
I pressed that a whole bunch of times. So I got this whole thing. I did a really short one. I just did 13 seconds. And if you press the plus sign, it's going to start looking like this. Did yours look more like that?
Yeah. Okay. Yeah.
Kinda.
Yeah. So for Jordan, yours looks more like this. So the little 2 mountain thing here, that's putting more of your record into the screen. So you can just go, right around there is probably good.
Okay, I'm going to try it right now.
And then you can see the shape of this right here. Each heartbeat. And each one of these points corresponds to those letters that are in the lab. So I'll bring that up. And I'll get to you in just a little bit, Jaden, to see what's going on there. You'll see that this right here, that first peak is the P, and then the Q is the one that goes down, and the R is way up here, and then the S is down there, and then there's a T. So it's P, Q, R, S, T. PQRST, PQRST. That's the name of the different parts of the heart rate.
When you open up the lab, it doesn't, like, I know it didn't show me those pictures at all.
Yeah, no.
What's that?
Those pictures aren't on, like when those instructions pop up, the pictures aren't there. It just has the description.
It just looks like words.
Yeah, it doesn't have any of the pictures where it says P or anything like that.
Okay, in order to see, in order to see this, should come up like that. You both have Macs, right?
I
No,
do.
I have an HP.
What's that?
I have an HP.
Because this is happening to me as well. When I go to the settings and it pops this instructions up automatically, it doesn't have the images in them.
Yeah.
And so I had to look up where to find the actual original files. So for Jaden, if you go into your hard drive, not your home account, but you go right into your hard drive.
How do I do that?
If you open up, like this is a new Finder window, like it just press Command N.
Okay.
And then this is my home folder right there, but way down here is my hard drive. Do you have the ability to go into your hard drive there?
No, I just have iCloud, desktop, and documents. You want me to show you my screen?
Yeah.
Oh, hold on. I have to wait until you're not.
Oh, sorry.
What the heck? Oh, it says host disabled attendee screen sharing.
Go ahead. Try it now.
Can you see it?
Okay. Okay. Up at the top of your screen, across the top, see where it says go? Click that, and then go down where it says computer. Click the hard Macintosh HD. Go to library. Go to application support. Go to LabScribe DL. Go to settings. Heart. And then this would be the rest exercise, HTML. Click that. I'll cancel that for a second. Yeah, cancel that. Yeah, not the BAK one. The one above that. Yeah, that one.
Can you see it?
What?
Yeah, it doesn't have any of the pictures.
Okay, try shutting that one down. You might have had it open already.
Okay,
Yeah, delete.
so...
Yeah, quit that whole, quit Safari.
Okay.
And then go, click it again over there.
Now click Safari again.
What's that?
Click the go and then computer.
Yep. Library. Yep, you're there. Application support. Subscribe, DL. Settings. Right. And it's the rest right there. Yep. Should. Please. Dang.
Maybe that's why we were so confused, because we couldn't see the pictures.
Yeah, I guess it's hard to do this without seeing those. Well, how about I just show you?
Okay, and we can just like write it down or something?
What's that?
Like, do you want us to write it down?
Yeah, like I'll show you what it is.
Okay.
So you don't have to worry about that. Because you really don't you don't do much of this lab, really. You just do the beginning. Okay, so give me my screen back.
How do I do that?
Up at the top, does it say stop sharing? Up at the top. Or maybe not. Maybe I'll do it.
There we go.
There. Okay. All right, so I apologize for that. This was a quirk, I guess. Let me look at the screen again here. So this right here, so you know to set it up like this. So you have the black one up there, the green and the red. And then exercise 1, it really has to do with when you get something like this, all you really need to know is the order of those letters. Okay. And so let me bring so this is my thing here. So
can you show us?
What's that?
Oh, sorry. Can you show us how to do the heart rate thing again? Because mine does not look like that still.
Mine.
To do this part right here.
Yeah.
That's where you locate the electrodes.
Yep, I did all that.
Yep. And then you press record.
Yeah, but how do you make your heart rate on the other screen on the lab scribe? Remember how you showed us the mountains? Like, which ones do we choose? Because every time I do mine, it just gets all messed up.
I think mine did too. It didn't look right.
Well, there's 2 buttons up here that change what you see. There's a bunch of, actually a bunch of buttons here. This button right here, it has a plus sign on it, will zoom in. Okay, and see that's very, very close. And then I can zoom out by doing this one. And I'm zooming out more and more. And if I zoom all the way out, it starts really looking crazy. And if you're doing 5 minutes, this is just going to be a blur. Going like that.
So you might have to do it a lot.
I'm sorry?
So we'd have to do it like click it more times if we have a 5-minute span instead of the 30 seconds.
Yeah, so you can get close enough. You click that plus sign close enough. And all the way out. The plus sign, actually the single mountain one too will do it. You see how it's getting closer? And now the whole thing, I'd have to scroll to see the whole thing. And as I get closer, it spreads out so that I can see a single heartbeat. Now, if we're going to look at the letters, P, Q, R, S, and T. Starts with this little peak. I'm going to look at this one right here. So the first peak is P, then it goes down to the Q, then up to the R, and then down to the S, and then up to the T. And that's the full electrical activity of a single heartbeat. So P, Q, R, S, T. Just the lowest point after the peak is the P, Q, R, S, T.
Do that one more time.
What's that?
Can you just do that one more time, the PQRST thing?
This first bump right here is the P. At the bottom of this right here is the Q. The top is the R. The bottom here is the S. And then the next little bump is the T.
Okay.
So you can see this right here, if it, just so we get the names, is T to T would be the interval between T's. Okay. The amount of time between that peak and that peak would be the T interval. From here to here is the R interval. Okay. So if I was going to look at I did a little table like you would do. So you get it at rest and then familiar and sedative and familiar and stimulating. The R wave amplitude. Okay, that's the first one.
Okay, that makes much more sense.
So the R wave amplitude, that's PQ. You go to the bottom of the Q to the top of the R. Right there. And we're measuring the strength of the signal. That's what I mean, we have amplitude, like an amplifier. How loud is it? So we want that. That number, the difference between this point and that point is represented, follow my cursor, over here. But if I press, that's the mean, that's showing me the average. If I click that once, I have all of these differences here. I want the second one minus the first one, and that will give me the change. So V2 minus V1 will give me the change between this point and that point. So the first measure of R amplitude is 1.399 millivolts. Now I'll put that right in as that for the lab, you need to do it 3 times and average them. But I'll just put 1.3. So I would put 1.399. That's the R wave amplitude. The beat period according to our instructions Looking for my website here. So that's the R wave amplitude. The beat period is the interval between two adjacent Rs. That's the really tall one. Keep that there. So I open up my lab again. I remember that's PQR, so it's from one peak to the other. So it's the R interval is right there. So I take the first one, I put it at the peak there. And then there's the second one. And now I'm looking at time. So this is showing me the difference that happens, but there's the time right here, right above it. The time that elapsed between this beat and this beat was 1.12 seconds. And if I did this one, look at that one, let's see if how much between these two beats. 1.14 seconds. Okay, so that second measure is between the two peaks. So if I went into my Word file again, I'll just put the 1.14, 1.14. And the P, now all of these are all intervals. So intervals, amplitude is the height, interval is the time. So we're going to be looking at the PR interval, QT interval, TP interval. Go back to the lab. So what was the first one there? PR. That would be P to R. So P, Q, R, S, T. Remember, so P to R, and it's up here again, 155 milliseconds. Do 3 of those and average them. Then you have QT. That's from here to here. That, so PQ, ST, so Q to T. You getting this? How it all, it's just, now the significance of this is each one of these parts of the heart rate corresponds to function within the heart, within, the opening and closing of valves. So when things happen to these parts, when these things go awry, that's how they diagnose heart disease or problems with valves or something like that. And in this case, we're looking to see if biochemically, because you were listening to music, it affected any one of those aspects of the heartbeat, of what the heart is doing, the controlling mechanisms of the heart. So each one of these, like what happens between the Q and the T is a particular function of the brain. It's of the brain, of the heart, the squeezing of the heart to send blood to the lungs, let's say. And that's what so we're measuring each one of those things. So in this case here, this is 355 milliseconds. So I'd put that in there. And what you're going to be doing is you do it as at rate, just continue here. I'll get to the average heart rate in just a second here. You do all these, then you do it again, and then you do it again. And all you're discussing is there anything that's changed? Did something go on average go up or to go down, and just leave it at that. Now the average heart rate is going to actually come from this. It's the only thing that we're doing with this graph on the bottom. Now to get the average heart rate, we have to go all the way out and see the full. So we hit that double mountain thing right there. And we keep on clicking it till it doesn't go no more. And now that's my whole record. And yours is going to look much squeezed. It's going to be it's going to look. And then you see this, you don't need to do this right now, but see the little magnifying glass that has the 2 arrows in it? If you click that one, it's going to shrink them down so you can see them. Because sometimes the thing goes it goes way up, and it'll squeeze them down so you can actually see them if these peaks are too high. Now, I want to get the average heart rate for this whole thing. And that's down here. You see my heart rate changed over time. It's low, then it's higher, then it's down, up, down, up, down, up, down, all that. What I want to do is I take these red lines and I put one at this end and then the other at this end. And there's my average heart rate. Now this right here, just like up here, I could press this and I have all kinds of different measures. Down here, I can click this and I get all kinds of different measures. And what you want is the mean. You want the average heart rate through the whole thing. You know all about means. We've been discussing that all semester. So that's giving me the average, even though right here, there's an incidental heart rate. Let's say the average is 54, but if I was going to look at that particular rate right there, that's like 61. And my lowest, well, I can actually do a range. I can do a minimum and max. There's the difference between my lowest heart rate and my highest heart rate. I'm not asking you to do this though, is 9.1 beats per minute. But I want the mean. The maximum was 60. The minimum was 51. The mean was 54. I'm actually kind of impressed with that. That's pretty low. I must be chill. So you're getting the whole thing. So there's a red line over here and there's a red line way over here. So you're getting the average of all of that. I would get, it'd be different if I could actually, I think I lost my other red line, went off the, can't find my other red line. Oh, well. So that's how you get the average. So mine was 54, that would go down here. And this is the one that I think you might be able to talk about. Maybe this one right here, it relaxed you a little bit. I would, brought me down to 50, and over here it was, 67, because I was listening to the different kinds of music. So this is a lot of moving the red lines around is what this is. And so you get 3 readings for each one of these. Just pick 3. Average them together, put them down there. You could cheat and just do 1. I won't know. I'll assume the average of 3. So you have all these readings, and then the discussion is simply, hey, I noticed that when I was listening to familiar sedative music, my PR interval went way down. That's it. That's all we're doing is just noting those things and learning to use the software to measure and to pinpoint areas of the heart rate. That was worthy of a movie.
Yeah.
So does that help? Is that clear?
Yeah, that makes much more sense.
Okay. Yeah, and not seeing the pictures obviously you're not going to get this if you don't see the pictures, and that's frustrating. These are all available on the web too, all the labs. are available on the iWorks website. And maybe I need to include those in case, in case the software is doing this, which it's, I'm not sure why it's doing that. It might be, and here's what can happen. It might be all those pictures, when you load up that file on here, all those pictures are actually on the web and it downloads them. And if they've moved them, that's it. Or if you're not connected to the internet, or you have a poor connection to the internet, it won't load the web page. You'll just get the text. And so stuff that we learn along the way. Okay, so that's lab 12. Does that sound good? And this is the last lab. So we're done with that device. And so you're free to, once you're all done with it, you can talk about bringing it back to campus or doing something. This thing. I got to send them back.